This is a project that actually Dr. Hilt and I collaborate on. So Dr. Hilt is another chemical engineering professor. And it's also part of our what we call the IGERT program. It's an integrative graduate education research training program that's funded through the National Science Foundation. And that program actually focuses on the interactions. It's called engineered bioactive interfaces and devices. And so what we're doing is we're looking at how biological systems such as proteins or cells interact with synthetic architectures that people develop. And so Dr. Hilt is really the synthetic architecture person. So he develops the hydrogels, and then we look at how those hydrogels interact with cancer cells, specifically for cancer treatment. So a little bit more about the project. We have hydrogels, which are really polymers. And a good example of a hydrogel are your contact lenses. So hydrogels are polymers that actually shrink and swell under different conditions, specifically temperature changes. And then what we do is we put iron oxide particles in those hydrogels. And the iron oxide particles can actually be heated from an alternating magnetic field, which is distance from the particles. And so the particles are incorporated in the hydrogels. We can heat those. And so the idea is you put the hydrogels with the iron oxide nanoparticles in a person who has cancer, and you can heat from a remote source. The alternating magnetic field will go through the skin, and so you heat these and you can actually kill the cancer with hyperthermia. The purpose of the hydrogel around these iron oxide nanoparticles are actually to first of all, to protect the nanoparticles and to protect the body from the nanoparticles. And also you can incorporate drug into the hydrogels. So it's you can do a combined chemotherapy and hypothermia treatment of the cancer.
Insert those into the body. Would you just like inject it, or?
So there's different ways that you can do that, and we haven't gotten to that place. All of our studies so far have been outside bodies and just growing cells in culture flasks and seeing how these things interact with the cells. If you would get to the point that you would incorporate these in the body, there's different ways. One that we have thought about is, if somebody has cancer, probably the very first step that they go through is surgery. And that's to remove the tumor, in the area where the cancer has grown. So during the surgical procedure, after you remove that tumor, you could stick these hydrogel systems into the area where the tumor has been, and that will help kill the remaining cancer that is in that area.
How long until you think that this maybe will be implemented?
It's probably down the road, because again, right now what we're doing is we're working in actual culture flasks. So we take cancer cells that we can buy from, there's cell stores called ATCC cell systems that actually sell cancer cells. And so you can grow those in an incubator, put them in a flask, and you can subject them to these systems to see what the cancer cells do. So we're in the real initial stages of this. So the next step would be animal studies. We're actually trying to find somebody over at the Cancer Center now that does some animal studies to start working with animals. And then it would go from there into humans. So it's pretty far down the road. A lot of our research is more basic research where we're trying to understand these things and how they work.
Are there any symptoms you foresee or you would expect after giving someone this treatment?
I think it would minimize the side effects that you see with chemotherapeutic agents, because it is more localized. So you can use less chemotherapeutic agent than what you would if you did a systemic injection. So I think you would see minimal or minimized side effects, if you would use this localized treatment.